Hi everyone, welcome to our summer experiment video series with Cornell iGEM. Today we will be going over how to extract DNA from strawberries. So what is DNA? DNA is a molecule or nucleic acid that carries important information for an organism. This type of information is needed for development, growth, and so on and so forth. Think of DNA as sort of a recipe or instruction manual for what it's telling your cells to do. So DNA encodes for RNA, which is another type of nucleic acid. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. RNA encodes for proteins. This sequence of DNA to RNA to proteins is known as the central dogma of molecular biology. Proteins are extremely important because they are responsible for how cells function, and they help cells carry out certain tasks. In iGEM, we can engineer bacteria to produce proteins that help us address the certain problems we'd like. DNA is actually very common. All living organisms have DNA, and we actually share a lot of DNA across all organisms. These are just some examples of the amount of DNA we share. For instance, humans and zebrafish share about 85% of our DNA. Today's experiment will be about extracting DNA from strawberries. Humans actually share about 60% of our DNA with strawberries. So in this experiment, we will be creating something that we're calling the DNA extraction solution. This will consist of water, dish soap, and salt. So the dish soap is important because it helps cause the strawberry cells to burst open, which allows us to actually access the DNA. We are using salt because salt helps separate out the proteins from the rest of the DNA, because the DNA is what we truly want. Also, we will be using rubbing alcohol at the end of the experiment, because basically DNA in alcohol is insoluble, which means that DNA cannot be dissolved in alcohol, which makes it so that DNA actually can clump together and is visible when we place it in alcohol. So when you're done with the experiment, if it worked well, you should be able to see this sort of white, almost string-like material on top that is actually our DNA. Stay tuned to learn more about how to actually do this experiment. If you would like another recipe, you can go ahead and go to this source here. Thank you.
Hello, welcome to another Cornell iGEM summer science experiment. So today we'll be extracting DNA from a strawberry. So first you will grab a strawberry and place it into a Ziploc bag. Make sure you seal the bag and also let out any excess air. So you'll start squishing the strawberry, and you want to continue to squish it until all the big pieces are crushed. Now you don't want to squish it too hard, as you'll damage the DNA and you will probably break the bag as well. So just be careful, and just remember to squish all of the large pieces of strawberry until it's liquidy substance. So now we will continue to make the DNA extracting solution that consists of 1 cup of water. You will also need 1 tablespoon of dish detergent. And you will also need 1 teaspoon of salt. Now, when you're mixing everything together, you just want to make sure that the salt is completely dissolved. The soap dissolves the lipids or the fats of the strawberry cell wall and nuclear membrane so we can access the DNA. Now the salt buffer solution, it breaks up the protein chains that bind around the nucleic acids so we can get the actual DNA. So we can isolate the DNA and extract it. Once everything is dissolved, then you can mix 2 tablespoons of the buffer solution, the DNA extracting solution, into the bag that contains this mashed strawberry. So after adding the 2 tablespoons, just make sure to reseal the bag again and start mixing the 2 liquids together by just squeezing the bag. Make sure that everything is thoroughly combined. So now once everything is combined, you can place a gauze or a filter over a plastic cup and just very carefully pour the solution that's in the bag over the gauze or the filter. You can also use a rubber band to secure the gauze over the cup. And it's also helpful to use a spoon to move around the liquid and make sure that everything is straining below. Once you've strained everything all of your solution, you can just go ahead and add an equivalent amount of alcohol. You can just eyeball this. And so now the alcohol is used to precipitate the DNA. The DNA is not soluble in ethanol or alcohol. You can either use isopropanol alcohol or ethanol. You may be able to get more with ethanol, but as you can see, there will be cotton-like fibers or wisps that form on the top. So that is the strawberry DNA. I hope you enjoyed this experiment. If you have any questions, please comment down below, and thank you for watching.